we are going to discuss another interesting topic, that is processing of mRNA in eukaryotes. So there are some basic differences between the prokaryotes and eukaryotes. One such difference is, in case of eukaryotes, both transcription and translations take place continuously, or which takes place in a cooperative manner. When few bases of RNA is formed, then automatically the translation starts, because the nature of the cell is different in prokaryotes and eukaryotes. In case of prokaryotes, you don't have something called nuclear membrane. There is no clear-cut nucleus. It's not a membrane-bound structure in prokaryotes. So this process is take place simultaneously. Whereas in eukaryotes, you know the structure of eukaryotes, where the nucleus or the nucleic acid chromatin, which is separated from the cytoplasm by a nuclear membrane. Okay, so the mRNA, what is synthesized in the nucleus, it will undergo some process called post-transcriptional modifications. So what is the product of transcription? mRNA. mRNA is formed after the transcription process, or otherwise called as transcripts. We call it as mRNA or RNA transcripts. So what happens before the mRNA is going to be transferred to cytoplasm? In case of eukaryotes, it undergoes some modifications. The modifications are referred as transcriptional modifications or post-transcriptional modification after the transcription. So it's called as post-transcriptional modification. Are you clear? It is also called as processing of mRNA. And one thing you should understand, this modification is specific for eukaryotic mRNA. You don't have such modifications in case of prokaryotes, because prokaryotic genome is somewhat simpler than the eukaryotic genome. Okay. So before getting to the different processing or modifications, let me tell you the structure of RNA, particularly the mRNA of eukaryotes. So this is the single-stranded mRNA. This is the product of what? Transcription. So any one of the strands act as a template or serves as a template for the synthesis of mRNA. The strand which acts as a template is called as template strand, and the opposite strand is called as coding strand. Okay. So this is the single-stranded mRNA, runs from 5' to 3'. This is mRNA. So within this mRNA, there are regions which codes for protein. How we mention, or how do we identify the starting and end region of the protein coding region? So you may also ask why it is not from the first base pair itself. See, in case of replication, in case of transcription, the process started from the whole sequence. So say for example, in case of replication, the DNA is synthesized right from the first base pair till the last base pair. Whereas in case of transcription, the one particular gene is going to be transcribed. That is also from the first base to last base. Got it? But what is here? So this is the mRNA. Actually, this is the product of DNA. So that means it is a gene product. Gene is transcribed into mRNA. You should understand that within the mRNA, this is not that the entire mRNA is going to produce proteins. It's not that the entire mRNA is going to be transferred to amino acids. Okay. There are regions alone, specific regions alone are going to code for protein. Such region is called as protein coding sequence. So how do we differentiate the protein coding sequence and other region? You have an mRNA. Within that mRNA, you have a translation start site and translation stop site. Translation start translation stop. அது எங்க இருக்கு, எங்க ஆரம்பிக்குது, எங்க முடிவு இருக்கு. Only that region of mRNA is going to produce protein. What about the rest of the regions? The rest of the region has got some other name. What is it? If the region which doesn't code for any protein that are located at the 5' end, they are called as 5' untranslated region, because this region is not going to translated. The same way you have some region at the 3' end, they are named as 3' untranslated region. So the structure of mRNA, don't get confused what is translation and all. Just understand the structure of mRNA. It has 5' UTR, it has got protein coding sequence, and then 3' UTR. That's all. Okay, what are the three important regions in an mRNA? You have mRNA 5' site, particularly the untranslated region, or simply we call it as 5' UTR. And then you have some region at the 3' end which is called as 3' untranslated region, which is called as 3' UTR. And the region that connects both 5' UTR and the 3' UTR is called as protein coding sequence. This is called protein coding sequence. Both 5' UTR as well as 3' UTR. And in between you have a sequence called protein coding sequence. It specifies amino acid sequence of the protein that will be produced during translation. So what is translation? The mRNA, the process of synthesis of amino acids using the codons of mRNA in the protein coding sequence. That is called as translation. The translation is recognized by translation start site and translation stop site. It would also vary in length according to the size of the protein. So you know that insulin that contains 2 chains, that too small of, it is in the range of 20 to 21 nucleotide amino acids, whereas hemoglobin is a larger protein which consists of 4 polymers. So tetramer in nature. So the size of protein coding sequence, the protein coding sequence would vary or depending upon the size of the protein. If it is a big protein, large protein, then the protein coding sequence would be longer. If it is a small protein, then it would be shorter. Okay, so what are the 3 main parts of mRNA? Eukaryotic mRNA. At the 5' end you have 5' UTR, 3' you have 3' UTR, and the region that connects the 5' UTR, 3' UTR is called as protein coding sequence. So this is what I was explaining in the first slide itself. The major difference between prokaryotes and eukaryotes. So in case of prokaryotes, ribosomes have immediate access to mRNA and translation begins on the nascent mRNA chain while transcription is still in progress. That is why I told the prokaryotic, in case of prokaryotic cell, both transcription and translation take place simultaneously. Once few bases around 10 to 12, 20 ribonucleotides are formed, immediately translation of RNA would also take place. But in eukaryotes, mRNA is synthesized in the nucleus first, and then it to be transported to the cytoplasm before it can be used as a template for protein synthesis. So the mRNA that is synthesized in the nucleus, from there it will be transported to cytoplasm. But before it gets transferred to cytoplasm, the initial products of transcription in eukaryotes are extensively modified. The product of transcription nothing but the mRNA or transcripts. So are extensively modified before the export from the nucleus. Girls, can you imagine the whole process? See, you have nucleus. within the nucleus you have double-stranded DNA. Double-stranded DNA la vandu oru edathila one gene is copied or one gene is transcribed, okay, by involving so many molecular transcription factors and eukaryotic RNA polymerase 2 and so on, and then synthesis mRNA. That mRNA is called as RNA transcripts or primary transcripts. This mRNA, before it is transported to cytoplasm, it undergoes some modifications. That modifications are called as post-transcriptional modifications. So let us see what are the different processing or post-transcriptional modification occurs. So as you all know, in prokaryotes no RNA processing at all, because nascent RNA is usually the mRNA. But in eukaryotes, the nascent RNA is called primary transcript. So one thing you should understand, prokaryotes there is no post-transcriptional modification. So what mRNA is synthesized, the nascent RNA is usually the mRNA. But in case of eukaryotes, the nascent RNA is only called as primary transcript. The primary transcript has to be processed. And then after the process is over, after the processing, it has to be transported to what? Cytoplasm.